Good afternoon, everyone. Let's read one more topic of biochemistry. So, as we say, starting tracking. So, topic of today: apoptosis. Pale on further slides. Before beginning apoptosis, let us have a small introduction about Unacademy. Unacademy is one of the largest platforms for the preparation of examination all over India, and you can get the best education at Unacademy platform. Here are the pictures of some best faculty an academy, you can get the benefit of the best faculty all over there for your examination. There are special classes, and in these special classes, you can ask your doubts and queries directly with the faculty member. They'll be very much willing to solve your questions, your queries, and you can have a one-to-one interaction so that all your doubts are. Then there are test series, and as I always say, it's important to evaluate how much you have learned so far. That is possible with the help of tests. Tests are conducted every week as well as they are conducted daily, and they are also based on the subject and topic which you read. You can make the most out of the SC and you can evaluate yourself whether you learn topic well or not. Then it is one of the most easiest ways of examination. There is flexibility, language can be of choice. There are many members in regional languages, unlimited practice. Like once you have done a quiz, the quiz as many of times as you want to. There will be no obstruction that no, you cannot do the quiz time for as many times as you can watch the video as many of times as you want. So unlimited practice, language can be of your choice and be flexible. Like if you are not in the town, you can attend the offline class, you can even go through the lectures recorded and you can channel and start learning. One of the most easy for the examination, then you will almost feel like a classroom, anything classroom. Because the best thing, start with your education. Like you can ask your doubts, they very much answering your questions, and there will be interactive polls. In these interactive polls, answer some of the questions, and you will also come to know how many students are opting for the options are thinking that yes, it is correct. And each and every option will be explained or indeed explained by the faculty member. So it's kind of interactive is a very interesting and learn anytime anywhere is another most important thing I've been starting that if you're not in the town or any time any of Unacademy. Then there is a feature with Unacademy that is the plus subscription. What is so good about this plus subscription? You can just very well take the subscription and you start preparation of your all the 19 subjects because all the 19 subjects are available at this plus platform and you can just go through the lectures and what is my personal suggestion is go for a go for one yearly or a 2 yearly program so that you can make most out of all of the available topic on the platform. You can make the most out of things, right? Otherwise, if you are going for a small duration like a month or two, then it's quite difficult for you to undergo all of the topics. What is my suggestion for a yearly or a 2 yearly subscription? The most out of it, and you can very well clear the examination plus subscription. Then there is one more thing. If you're going for plus subscription, my referral code that is FB1. Using my referral code FB1, getting an additional 10%. So for getting this 10% discount, my referral. So let's start with today's topic. The topic is apoptosis. right? So everyone knows about apoptosis very well. And what is apoptosis? So apoptosis, programmed cell death, programmed cell. So what is programmed cell death? Like when the cell is born, cell knows that how many number of divisions it have to undergo, right? So programmed cell death. Already the programming is done that it is going to live for these number of divisions. So the programmed death cell is also understood by another, that is definite lifespan of a cell. Definite lifespan of a cell. Right. Now this definite lifespan of cell actually read in terms of number of divisions. So the number of divisions will already be fixed. That is your programmed cell death. Now, if it is programmed cell death or it is determined number of division and the lifespan of a cell will be a particular definite lifespan. So how is it important? Why do we read apoptosis, or why it is so hyped up topic? How apoptosis important? How apoptosis is important? What is so great about apoptosis? All the unwanted cells are extra at a particular point of time, are deleted or out, or can say melted out from the body of apoptosis. Like for example, there are some of the who are born with a tail. That is like improper apoptosis in embryonic stage. Embryonic stage, proper deletion of cell does not occur, leads into kind of deformities. Tail is one of a kind of that kind of deformity. Apoptosis was not proper in the embryonic cells. Then another thing is presence of the membrane between fingers. Like we have got the three fingers, but in embryonic stages there are membrane present between our fingers. So these membranes need to be deleted, or the cells should die in these membranes so that now they can no longer further serve the purpose of attachment of the finger. The only place where the finger attached now will be palm, right? So this is how apoptosis is important. So this is one of the example, right? It is not only thing which I am telling you, but there are many places where apoptosis. In cancer, if proper apoptosis is occurring, not lead for the tumors. There are various cells faulty mutation occurs and which further lead to cancers. There is a proper apoptosis signaling won't change into cancer. Apoptosis has got its lots and lots of importance, right? If it is apoptosis, so how the cell is dying? The cell is dying means it is dying on its own, not because of some pressure or something. No one is coming and eating the cell also. It is a kind of suicide. The suicide. So it can be called as cell suicide. So whenever we are saying suicide, if death we are talking about a normal natural death, it means it is sudden. Suddenly death can occur. But if it's a suicide, it's never sudden suicide. It is like always decided kind of thing. So if it is suicide, it means it is pre-decided, programmed cell death, or it is pre-decided, right? It is the pre-decided thing. So this, let us take one another slide. So the cells which undergo apoptosis. So the cells which actually need to undergo this apoptosis, what will happen in these cells? What will be the major thing that will be happening in the cell? I will be dealing with whole pathway in detail also, but what is the gist of the story? Like what will happen? So in the cells there will be condensation of DNA. And when the DNA will condense, what will happen? The membrane will bleb out. Membrane blebbing will occur. and membrane blebbing occurs, it means there will be small vesicles of the membrane, small vesicles of the cells which is now into form. And this was your whole cell. From this whole cell has converted into the small blebs. Now, why blebs? Because there is no leakage. There is no leakage of cell environment. No leakage. Like in necrosis, there is a sudden breakdown of the cell membrane or nucleus. It is not a sudden thing. It is a pre-decided thing, right? So pre-decided. There is nothing like oozing out or anything. All the material will be inside these vesicles only. They'll be digested out and they'll be taken up by the macrophage, right? So reps, they contain all the cytoplasm, all cytoplasm and organelles as well, right? These blebs are the thing. There will be no leakage of cytoplasm, no leakage of cytoplasm or your organelle. This is the basic thing what will happen. Now we are saying that cells, they are undergoing the condensation of DNA, then membrane level will occur. So this condensation of DNA can also be called as nuclear collapse, or the nucleus it collapse. Nucleus collapse. Right. And because of the nucleus collapse, these blebs are also called as apoptotic bodies. Apoptotic bodies are formed. So this is the basic how it will occur actually, right? And it takes place through two pathways. There are two pathways. Two pathways. Whenever we are saying two pathways, in many of the other processes, we have seen these things which is called as two pathways. So what are these actually two pathways? What kind of things will occur? The pathways can be intrinsic and extrinsic. We are very well acquainted with the term intrinsic pathway and extrinsic pathway, right? So the pathways which are initiated in the body initiate because of the body, much better, because of the bodies. So initiated by body is intrinsic pathway. What is extrinsic? Initiated by environment. Initiated by environment is extrinsic, right? So intrinsic and extrinsic are the pathways. Intrinsic pathway and extrinsic pathway. So extrinsic pathway, we are saying, so what are the things that are included in extrinsic pathway? Extrinsic pathway, there will be first thing loss of nutrients, loss, or there will be less nutrients. So if there is less nutrients or there is less things which can survival of the cells, definitely it will try to kill cells which are actually not very beneficial, which are actually not working in a harmonious way. Nutrient loss. So it's a kind of cost cutting in the body. There is loss of, there is less financial support, then people will they'll start cost cutting those extra expenditures. So this kind of cost cutting, right? Nutrient loss. Then X-rays. All know the radiation are very harmful because these radiations they actually lead into mutations. So if there is a kind of X radiation exposure to the cell and it has undergone some kind of mutation, then definitely with that kind of genetic material, cell cannot further carry on its life. Otherwise, the life of the cell would be faulty one and it will lead into formation of unnecessary bulk of the cells which are not even serving the purpose. So these mutations, they are also, which are also because of radiation such as X-ray radiation into pathway of apoptosis. Then extra heat, so this is a thermogenic factor. This will also lead into apoptosis. And hormones, sometimes the hormones are disturbed because of the environmental factors. There will be unnecessary production of some extra hormones. These hormones can also lead into the intrinsic pathway. Why we have discussed extrinsic pathway here? Because about intrinsic pathway in detail, intrinsic pathway, and then we'll be dealing about the extrinsic pathways. Right? So let us move further to intrinsic pathways. Pathway. So this intrinsic pathway, what is the major basis of the conduction of pathway? Every action which is taken in cell is because of the genes or genetic material, genetic code. So intrinsic pathway is also in response to genetic code or genetic material. Genetic code will have a pre-decided lifespan of the cell. So this genetic code will decide that for how long the cell should survive or it should die. So this intrinsic pathway is because of the genetic code or the genetic material, right? And in this kind of pathway, mitochondria will play a very important role. Mitochondrial role which will be very, very important, right? So this genetic code is itself the reason of cell death. Intrinsic pathway, reason of cell death, as opposed to the necrosis. Cell death occurs because of the injury like mechanical damage or toxic chemicals or various other things. That is the reason. Here the reason of the cell death is simple, genetic code itself. There is no other reason but it is genetic code, right? So it is a kind of suicide because cell knows that how long it has to live and then how long it cannot now support the proper harmonious life cycle of the cell, right? Now some of the two, three terms which are associated, let's see them. So if there will be lots of apoptosis, it lead into atrophy. There will be lots of apoptosis leads into atrophy of a particular tissue, right? Like in cases of the autoimmune, unnecessary production of the molecules which are damaging the body only. Lots of apoptosis is leading into atrophy, and too little apoptosis will lead into cancers. If the cells which have got faulty genetic material, deleted genetic material because of various factors, if those cells are not eliminated out, then it will cancer. And if there will be extra apoptosis of the cells which are actually not needed to be it into the apoptosis stage, or the cells which we need for the survival, if they are entering into the death because of the apoptosis, is into atrophy. So one must try to remember these things, that if there will be lot of apoptosis, it will be atrophy, and if there will be too little, then it will be cancer. Right? So let us move further. Apoptosis is our programmed cell death, right? And this programmed cell death occurs in a well coordinated manner, right? So what are the events of sequence of the events of events occurring in apoptosis? What all will occur? So cell will collapse, will occur. Then there will be degradation of protein degradation will occur. Then what else? DNA will break, right? DNA fragmentation will occur. DNA fragmentation will occur. Then what else? The blebs which are formed will be unnecessary in the environment. They should be rapidly engulfed by the environmental cells or the macrophages. So rapid rapid engulfment of blebs by neighboring cells or macrophages, right? So these are the series of events, but much more would be happening at the molecular level, right? Other than the cell collapse, gene degradation, DNA fragmentation, rapid engulfment of blebs. Other than these events, there must be some other things also must be occurring. So we've seen all the things in detail, but before that, so where does this apoptosis actually occurs? Which kind of organisms does apoptosis? Apoptosis, it occurs in all the multicellular organisms. Multicellular organisms. In them, apoptosis will occur, right? So, how is it important? Did you see the thumbnail picture of apoptosis today for which we are doing class? Saw there was an ovum and some sperms were approached. So, this apoptosis, why did we select that special thumbnail? Because apoptosis is important from embryonic embryonic development till the senescence or till the death, right? This is the sequence that from the time the ovum and sperm they approach each other, and as soon as they divide into multiple cells, from that time some of the cells will be eliminated out and some will be kept from embryonic development till senescence. Where these apoptosis actually occurs in multicellular organisms, right? So multicellular organisms, then the apoptosis will occur, right? So we have seen almost the basic introduction about our apoptosis. Now let us move a little detail in apoptosis, that what apoptosis is and how the various factors they play the role of cell death, because we have seen various things that yes, this will happen, cell will collapse, DNA will fragment out, but actually which enzymes and which factors are responsible for this thing, this we should know. So we'll be starting with the heading, that is the biochemistry chemistry mechanism of apoptosis. So let's just write the heading, biochemistry and mechanism and mechanism of apoptosis. Right. So mechanism. So always remember the word associated with apoptosis is caspases. Word that is associated with apoptosis, caspase. So these caspases, they are not active all the time. They are actually present in of procaspase. Procaspases. Now, if they need to perform the apoptosis of the cell, they need to change active caspase, right? Procaspase, they are inactive, but they have the potential of changing into the dangerous caspase, actually now kill. So procaspase is an inactive form and caspase active form. So these caspase are the thing, once the procaspase has changed caspase will further form the thing. So what is the role of caspase? What is its role? The most important role of caspase is activation of endonucleases, activation of endonuclease, right? Endonuclease activated. Now, what is the function of endonuclease? Have read about endonucleases this time. That what is the function of endonucleases? What does it do? Endonucleases word itself is telling that it is a kind of diase enzyme and it will break down the right. It will break down the endonuclease break down DNA and endo. Two types of nucleases are there, endo and exo. Exo means it will cut the genetic material at the ends, and if you have endo, it will cut the genetic material somewhere in the middle. So breakdown of DNA at interior end, at interior places by endonuclease. So now if it is cutting in an active gene, how that gene would be active? No chance of its activity. So endonucleases will lead in the cutting down of DNA, right? It will degrade the nucleosomal DNA or it will degrade the DNA, thick DNA. Will degrade, degrade, degrade DNA, right? And this degradation, if you are saying it will degrade, so whether this degradation will be random or it will be specific, we are saying that these endonucleases. So have you ever heard about restriction endonuclease? Have discussed this in our class of rDNA technology. It is available on YouTube. Just go and search in our channel rDNA and by my searching you can get whole thing. So endonucleases they are very specific for a specific sequence. These endonucleases they are actually cutting the genetic material at a very specific place. It degrades the nucleic DNA at a specific place, right? And the degradation is not random, right? So we can say specific fragmentation, specific fragmentation, right? Specific fragmentation. So this specific fragmentation is also called as chromatin condensation, which we were seeing so long that chromatin condensation. So this specific fragmentation or chromosome condensation, right? Chromosome condensation. Fine. So chromosome condensation is occurring and specific fragmentation is occurring. So there was a PYQ that the endonuclease is working in apoptosis will break down the DNA at a specific place or the cutting is random. Many a times students they go for random because they think that in the end cutting has to be done. So it just needs to destroy the DNA. So once they need to be specific. But no, it is very important that it cuts at these places. So the question can be the role of endonuclease in apoptosis is specific fragmentation, random fragmentation, or any or none. So your answer goes to be specific fragmentation, right? Now, if you are saying specific fragmentation, so how much long these fragments would be? The fragments would be around 180 to 200 side long, right? And this is also called as internucleosomal damage. It's also called as internucleosomal DNA damage, right? Internucleosomal DNA damage. So the fragments will be 180 to 200 nucleotide long. multiple such kind of fragments will be there will be multiple such kind of fragments will be there. Right now, we are talking about fragments. How these fragments can be actually observed. These fragments can be observed with the help of agarose gel electrophoresis. Agarose gel electrophoresis. How would it appear on gel? How would they actually appear? Let us draw a slab. If these apoptotic cells are obtained from the body and then they are uh, undergone through DNA extraction and then actually led to run on the agarose gel, how would they actually appear? They will actually give such broad bands at the starting, and then the bands will become thinner and thinner. How actually bands will look like? Kind of the like these. Thickness of the bands will keep on going lesser and lesser as they are moving from the one pole to another pole, right? From one pole to another pole. So, thickness of the band. So, why we are actually talking about the thickness of the bands actually? Because this pattern is a specific pattern called as step ladder pattern. Step ladder pattern. And this step ladder pattern is because of what? Is because of the endonucleases. That is why the gene is broken down into small, right? So this step ladder pattern is in apoptosis. So whether the question is whether this step ladder pattern will be necrosis or it will not be seen in necrosis. So step ladder pattern can be seen in necrosis as well. So it is not a characteristic of apoptosis. Can be seen in necrosis also. So this seen in necrosis as well as apoptosis. But if they are saying that it is in apoptosis, then it can be its characteristic feature, but it cannot be its diagnostic. Like I just that it can just characterize. Okay, fine, this can be apoptosis, but it cannot be 100% said that no, this is apoptosis. Can be a characteristic, but one cannot go on diagnosing. Right now, if you are saying that kind of thing will be appearing in apoptosis, what about your necrosis? What kind of thing you will see in necrosis? So in necrosis, this is the gel color will be this kind of the patch of the DNA will be somewhere this kind of shape. So this is a kind of smear, isn't it? So for necrosis, the agarose gel electrophoresis give result in a smear way. So it is also called as smear pattern, smear pattern, and in process. Right? So always remember that smear pattern for necrosis and step ladder pattern can be seen in necrosis. Also, it's a characteristic feature of agarose that yes, it will always be there, but one cannot go on diagnosing it of agarose gel. Now, before beginning with the mechanism of apoptosis, all the cascades, all the names, let us have a look that when these cascades will be actually activated. What are those crucial points in the cell cycle where the apoptotic body formed and cascades activated? So whenever there will be misfolding of protein, whenever there will be of proteins, so the misfolding of proteins lead into activation of caspase. Misfolding of protein, activation of caspase. Right. So when caspase will be activated and these folding proteins along with the cell will be digested out. Now, if there is no activation of caspases, no no activation of caspase, what will happen? Caspase are not activated. What will it lead into? Lead into accumulation of misfolded protein, accumulation of misfolded proteins, right? And what will they do in the end? They will lead into various kind of cancers, will lead various kinds of neurodegenerative diseases such as Alzheimer's, Parkinsonism. So the elimination of these misfolded proteins by gas-phase is one of the important things to remember in apoptosis, because if there is accumulation of unwanted proteins, very much dangerous for the proper harmonious conduction of all the cell processes. So these kind of cells which have got misfolded proteins removed out, that now the cell can perform the all functions harmoniously. Now let us move further. Let us move to the mechanism of apoptosis. Mechanism of apoptosis. So mechanism of apoptosis, if we are saying, so mechanism of apoptosis will be actually done under two things. It will be and it will be execution or initial procedure. Activate and in procedure every degraded out. So mechanism of apoptosis can be dealt under two heads, that is and another is carrying it forward or execute can be the one for this, or can say actual gradation, actual right and actual. So if we are saying initiation can be because of intrinsic pathway, because of the body's mechanism, or it could be because of extrinsic as well. Initiation, right? It could be intrinsic or it could be extrinsic. And when we are saying that actual degradation, so in actual degradation, your genetic material will be actually degraded out with the help of endonuclease. So actual degradation will occur with the help of actual endonucleases and caspases. The caspases are responsible for execution or actual degradation 3, 6, and 7. Caspase 3, 6, and 7. And they will lead in activation of endonuclease and degrade, right? Now, if we are, this is the execution part, we'll be that also. But when we are talking about initiation and an intrinsic pathway and extrinsic pathway, the caspases which are responsible for this, let us talk about them. So, the caspases which are actually responsible for initiation are caspase 8, 9, 10. Caspase 8, 9, and 10 for initiation, caspase and 7 for the actual, right? So intrinsic pathway and extrinsic pathways. Caspase which is active. So the caspase which is responsible for intrinsic pathway is our caspase number 9. Caspase number 9. And also remember that all the case caspases are actually present in procaspase form. Are present in procaspase. Now for extrinsic pathway, the caspase which is responsible for caspase 8. But can you see here that we have written caspase number 10 also? Why this 10? We are saying pathway occurs with 9 and 8 and here 3, 7. Then why that extra 10? There's a twist in the story, that is caspase 8. Caspase, caspase 8 is lower organism. Lower organism. Then for organisms like humans, which caspase is there? This caspase number 10. Caspase 10 is for humans or a human caspase, right? So these all things are very much important according to the competitive exam point of view, because you should remember the number of the caspases and which caspase is present in one, which caspase is present in lower organisms. These all things will actually matter uh, how you are attempting the exam. So you remember the name, that what is 8, what is 9, and what is 10. So right, we are good. Now that we have understood numbers of the caspases and we have already manner, so that you can just note down this slide, that you will remember the things well. Let us move. So these apoptosis caspases are actually present in the form of their procaspases. They are not actually directly caspases, but they are the procaspases. So, these apoptosis is very important for limiting cancers. So, the proto-oncogenes are the actually units which regulate the whole mechanism. So, we will be taking the story from there and we'll further, right? So, proto-oncogenes, proto-oncogenes. What are these proto-oncogenes? Proto-oncogenes, if they are converted into oncogenes, they will lead to cancer. So these proto-oncogenes themselves, they are divided into categories. And the two categories are pro-apoptotic, pro-apoptotic and anti-apoptotic. Anti-apoptotic, right? Pro-apoptotic and anti-apoptotic. That is another color. So these pro-apoptotic genes, what will be function? Function will be to promote apoptosis. Apoptosis. Now, what will be the function of anti-apoptotic gene? Function, as the name itself is saying, anti, will be its function will be to stop apoptosis. Stop apoptosis, right? So there are some of the genes actually fall in the category about them. We will be talking about them. The of the genes are actually present in the category. So the genes which are present in the apoptotic gene family are BAX, then BAC, then LXS, then BH3, only protein family. Only protein family. And this has got its further divisions. Next slide, about BH3. Then about the anti-apoptotic gene, what all anti-apoptotic genes are there? BCL2, then LXL and MCL, right? So here there is a thing that this BCL-XS and BCL-XL, BCL-BCL you can. So how can you remember that which one will be the pro-apoptotic and which one anti-apoptotic? So S is for to stop cancer. Stop cancer. So they are pro-apoptotic. It will lead into death of the mutated cell. And L L is for linked with cancer, with cancer, because they are not letting the cell to die, the faulty cells. They are not dying because of the anti-apoptotic gene, right? So the things which are falling in the category of pro-apoptotic genes are BAX, BAC, BCL excess, TH3, and those who are falling in the category of the anti-apoptotic genes, they are falling in the category of BCL2, LXL, and NCL2. Right. Now, let us take the story from BH3. H3, H3. They are the only protein family, are the protein family. Right. So, they are present on surface, they are present on cell surface. What else about them? They are the stress sensors. They are the stress sensors. Now, these are themselves divided into groups, as I told you. So, the two groups are BIM, BAD, BID, and Uma and Noxa, right? So, BIM, BAD, and BID, Uma and Noxa are the protein family of pH3, right? Have understood about all of the genes, all of the proteins. Let us move further how the actual process are. What is the actual pathway? So, these all we are reading under the pathways, right? So, the first is intrinsic pathway. Intrinsic pathway. This intrinsic pathway, also called as mitochondrial pathway, also called as mitochondrial pathway, because mitochondria will play the most important role in cell death. It is also called as mitochondrial pathway. And along with this, also the major pathway is the pathway. And when will it be activated? Activated in stress and injury. Stress and now let us talk about a normal cell and then compare it with apoptotic cell. If this is a normal cell, then when we are saying that it is quite related to mitochondria, so drawing a mitochondria, because when we are saying that mitochondria plays a role, let us in the context of mitochondria. What happens in mitochondria? Mitochondria. So in mitochondria, there will be certain things will be attached on the inner membrane of the mitochondria and outer membrane. So the things that are, let us draw some of the things which are present on the inner membrane. Let us draw some on the outer membrane, right? So the one that is present on the outer membrane called BCL2. And BCL2, if you can just remember the last slide, it belongs to the category of the anti-apoptotic gene. It is on the outer membrane, on outer membrane of mitochondria, and it is anti-apoptotic, right? Then these things, these, what are they actually? They are your backs and back. And we know backs and back, what is their function? They are actually apoptotic. They are apoptotic in nature. So those which are apoptotic are in the inner side so that they are not exposed and they are not leading to apoptosis. And those which are actually anti-apoptotic, they are outside. So the balance is quite there. The cell is not undergoing any kind of apoptosis. And now, one more thing is present in cytoplasm, that is called as IAP. What is the full form of IAP? This inhibitor of apoptosis, inhibitor of apoptosis. So, two of the things which are inhibitor of apoptosis are towards the cytoplasm, is in the cytoplasm, so it will inhibit the apoptosis. There are two things, BCL2 and IAP, which are actually now limiting the apoptosis, which is actually stopping the apoptosis, L2 and IAP. So the balance is there. The structure of what the normal cell is a normal cell, non-apoptotic cell, which is harmoniously living. This will be the condition of a normal cell. Now let us move and see how the apoptotic cell will actually look like. What will happen in the apoptotic cell? Is there anything different from the normal cell in the apoptotic cell? So let us move towards the apoptotic cell. Let us draw a cell, and most importantly, let us draw mitochondria. This is our mitochondria. Now, what is present on the surface? On surface, there will be H, and H3 serves as the stress sensor. Stress sensor, right? And what other things are present on this? There will be certain things which will be present inside the mitochondria if the cell is in the harmonious way. The things which are present inside the mitochondria, they are cytochrome C. We know that cytochrome performs the electron transport chain. So cytochrome C, so its normal function, and it is inside the cell, its normal function is to perform electron transport. When we are talking about apoptosis, its function will be different. It will lead into the formation of apoptosome. Let us see that also. What is this when it is inside and it is going to come outside? That is the thing. So it is called as SMAC dihydroprotein. SMAC dihydroprotein. What else will be present? What else will be there? So we know that the interior of the uh, mitochondrial membrane, it also contains backs and back. So let us draw backs and back, backs and back. Now we also know that on the surface there is a BCL2, L2. So we have drawn everything. Now let us see how the whole process will actually occur. Right? So in apoptosis, what will happen that the backs and back will be expressed on the surface of the mitochondria. They will just come on the surface of the mitochondria. Backs and back. If they start expressing on the surface, what will happen? They form a kind of tunnel. They will lead to formation of a tunnel, right? So they will be expressed on the surface of mitochondria, and because of their expression on the surface of mitochondria, the mitochondrial membrane will become permeable. The outer membrane will become outer mitochondrial outer membrane will become permeable, right? So if it will become permeable, these all things which we have specially mentioned, that is SMAC dihydroprotein inside the mitochondria and cytochrome C inside the mitochondria, they will come out. So two events occur, and two things, or can say two things will occur. That is mitochondrial cytochrome C will come in the cytoplasm, and when it will come in the cytoplasm, it function of each get changed. It will become cytosolic cytochrome. Help of this channel it will come out, called as cytochrome, cytoplasmic cytochrome C, and its function is to form the apoptosome. And another thing that will come out in the environment will MAC diabeloprotein. It will also come out. So the cytochrome C, along with there is one more thing that is present in cytoplasm, that is this. Name is apoptosis activation factor 1. So now cytochrome C and apoptosis activation factor 1, they both will combine and will form apoptosome. Apoptosome, right? So what will be the function of apoptosome? The function of apoptosome will be to activate the procaspase, and procaspase will convert into caspase 9, right? So this is how the story will occur. And when caspase 9 will be formed, it will be activation of endonucleases. But in the last picture we saw there is one more thing in the environment, that is IAP. IAP, right? This IAP will always try to inhibit the formation of apoptosome. Will always try its best that this apoptosome should not form. And here we saw that the backs and back, they will lead into formation of the pore. So if there will be a sense of stress by BH3, give its signal to the backs and back, and when it will give its signal to backs and back, they will be activated, start expressing themselves on the surface. And this BCL2 is anti-apoptotic, so whenever there will be stress, it is inactivated by BH3. This is how the process occurs. So let us make a very quick flow chart of the whole thing. See, BH3 will sense stress, and back plus backs will come on the outer membrane of mitochondria, and it will form for channels in outer membrane of mitochondria, and because of that cytochrome C and MAC diabloprotein will be leaked out. When cytochrome C will come out, will combine with apoptotic activation factor 1, and it will formation of apoptosome, and apoptosome will activate the procaspase and convert the procaspase into caspase 9, and it will further activate the endonucleases. How story goes, how the story goes in intrinsic pathway. You can just note down this flowchart and you can well remember the things. Now, the another thing, that is IAP. IAP, it inhibits the apoptosome. So what is the function of SMAC diablo? SMAC diablo protein will block IAP. Will block IAP. The function of SMAC diablo to block IAP, because IAP will block apoptosome, but we don't want it to happen. SMAC diablo is for controlling IAP, right? I hope you have understood this pathway well. Let us move towards the extrinsic pathway. Extrinsic pathway. This extrinsic pathway is also called as depth receptor mediated pathway. So this is like untimely death, intrinsic pathway. So the important death receptors in the body, there are many. We'll be talking about two. The two are NFR and FAS. Mostly FAS based are asked in exams. NF is tumor necrosis factor receptor, and FAS, it is a marker for the FAS 95 is a receptor of fast death. Receptor is a marker for the fast death receptor, right? So the two markers or the two death receptors in extrinsic pathway are TNFR and FAS-R. Let us move further that how the things will occur in extrinsic pathway. Let us take a slide. This is the cell. If this is the death receptor or your TNF or FAS receptor, in generalized term, we can call it as death receptor, right? So it will be present on the surface. There will be another thing which linked with it, that is adaptor protein. Adaptor protein. And this adaptor protein further linked to caspase 8 or 10. If we are talking about humans, it is caspase 10. Think like this. So how the flow work will occur? Let us take another color. If there will be a ligand coming and attaching the fast receptor and it will be giving it a signal, then what will happen? The adapter protein will be activated. The ligand will actually activate the adapter protein, and this adapter protein will in turn activate the procaspase. Procaspase change into caspase 10, the activity 1. Right? Now, the extrinsic pathway is quite simple because just a ligand should come and it will change structure of adapted protein and adapted protein which are linked to procaspase activate caspases and it will further lead to degradation. So this was about the extrinsic pathway in extrinsic pathway, and that too initiation, right? For execution or further actual degradation for both the pathways, intrinsic and extrinsic, it will be same. So let us move further. That is execute. How the execution will occur. The caspase 3, 6, 7 are responsible for execution, and when they will be activated, they will activate endonuclease and they will feed into chromatin condensation and fragmentation. Right? So like this, if the genetic material is broken down, it means that is the end of the story, end of the life of the cell. And as the life of the cell is ending, our session is also ending. That the genetic material is fragmented out, nothing more can be talked about because if genetic material is no there, no more present, no more in a viable state, what else can be done? The boss is dead, workers cannot work without them. The genetic material is over, story is over. How? Execution occurs very simple. Intrinsic pathway as well as execution, both of the things are very simple. We are talking about apoptosis, right? So execution 6, 7, right? So like this, we are done with one more beautiful topic of biochemistry, that is apoptosis. Like my video, hit the like button, give me a thumbs up. It really motivates. And please subscribe our channel, that is Let's Crack NEET, and hit the bell icon so that you can get all the notifications, all the upcoming. Lastly, don't forget to use my referral code for your plus subscription. Really will be helpful for the preparation examination, and you can get an additional 10% discount by using my referral code. So don't forget to use my referral code, and keep on studying, keep on enjoying. Thank you very much.